So next up is Bruce.
Great. Thank you. Thank you very much for this opportunity. You asked for comments on some of the things that you covered earlier, Janice, and after having spent 10 years working on a single species of orchid trying to propagate, augment, and reintroduce your 5-year timeline, it's going to be very difficult for some of the species that we're dealing with. I know for Pseudopodium, you've probably been working, well, since 2008 or something like that as well.
Yeah,
so,
something like that.
yeah. So I think the epiphytic orchids are going to be a challenge to in that sort of timeline. But anyway, it's also wonderful for me to hear, and I hope to learn more about some of the techniques that you're using. During our process, we tried seed baiting and mycorrhizal isolation, and we just are striking out on some of those things. So the workshop that, some of those other workshops Jenny was talking about sound really of interest to us, so thanks. Well, back in 2006, we were approached by Everglades National Park and the Institute for Regional Conservation to look at this protection and augmentation of rare plants in Everglades National Park. This was largely based on the work that George Gann and collaborators did, looking at the endangered species of South Florida. And they wrote a huge book, very useful, lots of great historic information. And from that, we worked together to kind of narrow things down to species that we're of interest to us at Selby Gardens, which are epiphytes and ferns, and then to begin work on those. So, of course, they were alarmed. The park was alarmed about the alarming loss of native plant species, 8% extirpation, 17% critically imperiled, and mostly in the last couple of generations. And the focus was a lot on Long Pine Key. This is an upland area in the park, and there are a lot of the rare species occurred in this area. So that was a lot of our focus right in here. So some of the last remnants of rock pineland vegetation and a lot of other very interesting flora occurs in that area. So. Of course, you all know these goals. This is a talk I gave to my members at Selby Gardens, so I've got some kind of goofy things in here, so just bear with me. But these are the goals that we set forward to begin this project. And we narrowed it down to 8 species of orchids and ferns. We were interested in the bromeliads too, but we didn't really identify any that were in need of augmentation at that time. And then a couple of them are amazingly rare in the park, such as Lomeriopsis kunziana, the holly vine fern, Trichocentrum undulatum, and some of the extirpated plants. We also made an effort to try to find some species that hadn't been seen for quite a few years, like Abrasia caudata, Macrodenia lutescens. But there is a population of Trichocentrum in southern part of the park, about 500 individuals left. Well, let me just go through some of the challenges that we faced. This was a huge learning curve for us. This was the first applied conservation project that we undertook in Florida. And maybe that's why it took 10 years to get where we are now, because we're really trying to do things ourselves, and we didn't have a lot of advice at the time, although Scott Stewart did help us. So some of the challenges, we were talking about exotic species. You know, Brazilian pepper, this was kind of fun because I had these 2 Brazilian students come to visit. They had never seen this plant before. So I handed it to them and told them what it was. They had heard about it, but had never seen it. So it's the national tree of Brazil. Yeah. In access down in that part of the park where some of these species grow is a big challenge, just getting through. And it seemed like all of the Trichocentrum only grow in places where there's manchineel. So you had to kind of push through those to get to the capsules. And then the cultural challenges that we faced, this Galleander, we can never get it past this stage. We can never get it to develop leaves. So we're doing a few things wrong. When we went to look for the Brassia, there was this guy, he heard about our search, so he sent me this dot. This is Big Cypress Swamp, and it says, General Brassica area, I think. He had seen this working on a road crew about 15 years ago, and he remembered he was absolutely certain it was a Brassica caudata. So we went out there. It was a great trip, a lot of fun to go explore. We didn't find it. And then ecotourism. For orchids, it's a big deal. A lot of people go out. Even these orchid clubs they try to work to do some protection, but the trampling in this part of the park was pretty severe. And of course, poaching and extraction is probably the reason why the trichocentrum was extirpated from the northern part of the park, where the public has pretty good access.
Say that again. I mean, because of that book?
Because of what?
You think because of that book, or just because of the ecotourism?
The ecotourism.
Yeah.
Yeah. And then collectors. Because the trichocentrum is really a magnificent orchid, big, beautiful sprays of flowers. So, but there's certipodium. So there's a number of orchids that were extracted for a number of different reasons, including collectors. And then habitat alteration has also been a big impact, of course. So, and during the time we were down there making a lot of trips, we noticed very conspicuously 2 capsules, one from a certipodium and one from a trichocentrum went missing. We're pretty sure, and it was after that, one of those 2 orchid tours. So, but the real challenge, the real faunal challenge, is this little fly that, well, you'll see the damage, but we were getting almost no capsule production. The first year we went down, there were about 500 individual plants in Coots Bay in South Everglades. And the first year we started looking, we found 2 capsules. So the park didn't think that, they thought we should leave them both that year. But the reason is this fly, well, poaching reproduction limitation, and here's some of the capsules. It's a big, magnificent plant. The leaves can grow, 50, 60 centimeters long, and then big sprays of flowers. So in 2006, there were the 2 capsules, and it's exactly because this fly infests, it lays its eggs in developing inflorescence, and the larvae mine the stem, and they pupate inside the stem, and it basically aborts the flower stalk. So. But the problem is, the fly's probably rarer than the orchid is. So any thought of, we kind of joked about going and spraying, but because we wanted to get a couple of capsules to work with, but obviously we couldn't do that. So here's some of the damage you can see with the completely collapsed flower spike. Occasionally, they'll send out a lateral branch from the base if the damage isn't that severe. So there can be some flower production, but it's very, very limited. Closer view of the oviposition wounds. And here's the larvae actually mining, pretty severe damage and pupating. So this all happens between about January and March. The plants begin to bloom, the fly comes in, does the damage, and nobody knows what the fly does the rest of the 9 months of the year. That would be a really interesting project for somebody to look at. We don't see the damage on any other species in the park, or even in cultivation. It hasn't been observed. So we went in and did an intervention, and we designed these exclusion bags. My wife actually sewed those, and we had about 5 of them, and we tied them around the plant, and you can kind of cinch them off at the bottom to prevent any fly from getting into the plant. And we actually got the next year we got 9 capsules. We did a little bit of hand pollination as well, but that seemed to work. So we left the bags on until the flower spikes were strong enough to resist the fly from ovipositing the eggs. See the cotton batting at the bottom to make sure that they didn't land and then crawl in as well. So we put some tags on these things to tell people that were going in to look at the plants what was going on. But we got some really strange reports back about some odd contraptions being used in the park. So it was pretty funny to see the reaction from visitors. It's not in an area that you can easily get to. You got to hike in a little bit through across a canal. So then after the tissue hardened, which was about 3 or 4 months later, we went back in and took those out. And as I said, we got 9 capsules in 2007, 2008. One we took, so we collected 3. One was to be used for using a banana paste, mixing the seed in with banana and spreading that on the trees. We don't think that's a good idea. I think the raccoons really liked it, though. Or if you do that, you were talking about, I think, spreading some seed as well, using natural seed. In hindsight, we should have put some like wire mesh over that to protect that, but we haven't taken that any further at this point. And then so we kept some for propagation and for storage. And since we were not able to isolate a mycorrhizal fungi, we decided to do asymbiotic orchid propagation. And so we had, you're talking about all the different kinds of people that are involved in these projects. That was a real eye-opener for me. Students, volunteers just really took this on and did a lot of work. But what was also surprising, when we first started this, we thought, we're going to do this and wrap this up in 3 years. That was kind of our timeline, and that was such a big mistake. First to get the capsules and to grow the plants and do all the work, get them hardened off. And another really big issue I was talking to Dennis about is in the national park and probably other federal whole lands, we were not allowed to keep any of the plants for even ex situ propagation and preservation. We either had to return all the plants to them or destroy them. So that's in Everglades National Park. I don't know if anybody else has worked down there on these sorts of things. They're very, very adamant.
What was their justification for NAOCC collections?
It's the law. It's a federal law that park property.
I see.
Yeah.
It belongs to the park.
I understand.
Yeah.
And so to keep track of it, they need to either follow it or display it.
Yeah. So we grew a lot of plants, way more than we needed. We didn't really know how many we would need at the time, but we ended up putting out about 220 plants over a 2-year period, and that was in July in 2011. So you can see the plants are doing pretty good, but those are 4 years old at that point. So, and these probably aren't going to bloom for another 5 or 10 years. It takes a really long time for these to bloom, but it's a great place to work. Beautiful tropical hardwood hammock forest, lots of epiphytes. And this was the habitat where the plant was found by Small I think in the 20s. And there's a photograph of them in this hammock that's just magnificent. The plants, the leaves are this long. And what's interesting is that where the species grows now in Coots Bay, they're in Buttonwood in very bright, sunny locations. So entirely different sort of habitat. But since the park had the historic photo, they wanted to get it back into the northern part of the park. Hopefully far enough away from the fly also that it won't be a problem. Of course, I have to show my members, my volunteers, what it's like out there in the summer. It's fun. I love that. So we, as far as putting the plants out, we had just one photo of them pretty high in the canopy, but we thought we'd try them at all different levels. So we started pretty much near the ground on downed logs and moved all the way up as high as we could get with ladders. And we used, I think it was maybe a bit of a mistake, we used foreign materials, shade mesh and grafting tape to hold them on. We put a little bit of sphagnum, but the reason is that we had to go back in and remove all that stuff because the park is very adamant about not introducing any foreign objects. So, but we were getting pretty good attachment after a couple of years, and well, we also found that just tucking them in on horizontal branches underneath resurrection fern was as effective as tying them on. So here's our group. You need a really tall ladder down there. So here's 2014. This is the last time that we went out to do any monitoring. We're going to go back pretty soon, Craig, right? Yeah. Check out things. But you can see the ones, here's one right there, nestled in. It's colored up nicely because it's very high up in the very bright sunlight. Another one here, and this kind of mess. But the attachment is pretty good. We got about 30% survival. So they're about, I forget how many plants, but it looks like a really successful outplanting. We're just not sure how long it's going to be before they flower. We put plenty of plants well off the trail so that, because it's in a pretty public area in part of the park where a lot of visitors go, but we intentionally put some also in branches overhanging the trail, so it's just going to be a beautiful display, hopefully, when they bloom. So a couple of other species that we worked with, we were not successful with. I mentioned the Galliandra. It's actually bicornuta is another name that's used. We just couldn't get it to differentiate on the auger. And also Oncidium insatum. This one we were successful at propagating, but in the end, the park decided not to do outplanting with this. They felt the population was already good enough. We also worked with a couple of ferns, as I mentioned. Unsuccessful for the most part. Because of the areas where we're putting these things back into are so remote that there's nobody going to go out there with a watering can on a regular basis, and the ferns need that. But so we're still looking for a couple of species on that list. I mentioned the Brassia and the Macrodania. So if any of you come across that in your travels or hear of a locality, we're willing to try to track that down and get some capsules. So a lot of thanks for a lot of people involved in this. You can see young Scott Stewart there on the right. And the IRC was really a main partner in this and did a lot of field work with us. And here's Jimmy Saddle is the park botanist now and a very good collaborator as well. So that's our foray into reintroduction effort. And we have the interest and some capacity to do more, so that's why I was really anxious to come down and meet everybody and talk about these things. Thanks.
So, do you think, so when we were spreading seed the other day, I had this, substrate idea in my head too. You mentioned using banana, and I was thinking, it'd be good to have some way to get this seed to stick. Yeah. And banana didn't cross my mind. Potato did. And I'm wondering if the banana was maybe if you use something more.
Yeah, we've talked about some other sorts of things, but to get more capsules would require us to go back and do another 5 years of.
Yeah, no, I'm just thinking for other projects,
I think it's
some wet down some Hungry Jack flakes or something.
Yeah, right.
That potato might be a good idea because you can use that with your auger to grow things. Might work.
Yeah.
Why do you think the Match-Neal site seemed to match, or what would be the similarities of Match-Neal sites versus out of Buttonwood stuff?
Well, the Manchineel grows among the Buttonwood. So that's all together. The Trichocentrum, the Buttonwood, and the Manchineel all together. But it just seemed like every single Oncidium that we were after was surrounded by Manchineel.
But the hosting it was Buttonwood?
The host is Buttonwood. Yeah.
Any other questions? All right, so Nick, are you going to? Naples is on.
So I don't have a formal presentation, so I'll just kind of keep it short and sweet. So my name is Nick Avey. I'm the Associate Horticulture Director here at the Naples Botanical Garden. So I definitely have an interest in orchids as well, and I think as a garden, we are seeing that potential come to light as we've been doing a lot of expansion in the garden. For anyone that hasn't been here recently, you've seen all of our expansion we're doing currently and a number of things coming up in the future as well, including our new Florida garden that we're putting together this summer. But within that, we've definitely noticed an increase in interest and popularity of the orchids. And it seems like the more orchids we put out into the landscape, the more interest there is, and it's just one of those things that just is a highlight of our garden. And I think there's a real big interest for it within the community from local people, local orchid societies, as well as people coming from out of state, from up north, wanting to see the plants growing in a semi-natural to natural habitat. So it's I think this garden has a real opportunity to kind of showcase and advertise orchids to the public. Definitely an interest of mine also is the native side. So it's pretty fascinating to be able to have a native population of orchids that we can advertise and be able to show people, showcase. And one of the advantages we have in the garden, a number of advantages, but we are open year-round. A lot of these plants we can showcase in either somewhat natural site within the garden property or even on some more natural areas out in about 170 acres. So we have a number of habitats out there we can utilize. We have a pretty good amount of people. I don't know what the last numbers were on how many we had come through the garden last year. 100-something thousand? Almost 200,000? 240. So you can imagine that's a lot of eyes coming through, and sometimes they're plant people and sometimes they're not, but being able to open people's eyes to these things is a huge plus for the garden. And we have, since we've done our new expansion, we have a dedicated orchid garden now. So we have plants that are maintained mostly through our volunteers. We have a small orchid converted shade house. It's now an orchid house. We have our orchid collection. We have a group of volunteers from the Orchid Society that maintains the orchid greenhouse and helps us with the displays and on the public side of it. And so we are able to have a rotational display of flowering orchids as well as ones that are established in trees. And then in addition to that, we have the rest of the garden. Of course, we have orchids trying to stick to the natural themes of Brazil species, Caribbean species, hybrids that might come from certain regions or in those areas as well. And then looking into our natural areas, we have some of our native Florida plants. So we have a naturally occurring population of Encyclia tempensis, so it's definitely one of the more common native orchids, but we have a thriving population in our natural preserve areas. And I've been able to, on a small scale, experiment a little bit. Last summer, I guess, at GCU College, which is what we're in here, there's actually a laboratory right here, and we asked if we could use some of their laboratory equipment and start to just see what we can do with propagate some orchids. So we were able to use the equipment, autoclave, and a hood. And on a small scale, we've been starting to propagate orchids on our own and focusing pretty much on native plants. And so that's kind of spurred more interest. And so I think it's about last fall or summer into the fall, we met with Larry Zettler, and through him, Michael Kane, as well as some from Atlantic Botanical Gardens as well, we've gotten some other plants to put into our natural areas. And I think we have this really unique location here, being as that we're surrounded by corkscrew, fat-cahatchee, panther. We've got all these amazing habitats we're surrounding, and we're kind of right in the center of it. We have a very good representation of different habitats out in our natural areas where it's very accessible, very easy to get to. So we have a very good testing plot for putting some of these orchids out and just kind of seeing testing methods, easily being able to go out and see what we've done right, what we've done wrong. So being able to experiment with some of these plants that we grow on site, as well as ones that we've gotten from some of these other institutions. I think we have a really unique spot here in being able to do some of these things. So basically at this point, I think we're kind of still in a little bit of an exploratory mode here, just kind of figuring out what our role is going to be within this group. But I think we have a very strong presence here in Florida. We're expanding quite a bit through our experimentations with the outplantings and small propagation we've done. I think we're pretty excited, and we're really just beginning. And I think there's a lot of interest, and we have some funding that's come in already through some fundraisers that we've had. So we'll be able to figure out in which direction we're going to go with the funding that's come in. And other than that, we're just excited to be part of the organization here.
Okay.
Any questions for us?
I remember reading some things a while back with David Fairchild's books. The fact that he brought things back to the country, and he started saying things like, growing a little faster than expected, right? Are there any contingency plans for you all if you see something growing a little more greatly, expansively than expected? I mean, you can do a little control, removing capsules or things of non-native species?
So as far as, yeah, the non-native orchids that are
Correct.
around, yeah, and certainly that's, would be true for orchids as well as any other
Correct.
plants that get brought in. We very actively monitor any plant that comes in and kind of notice or look for any kind of invasive potential. And once something is seen, it's usually removed or isolated and or removed from the property if it looks like it has that potential.
So have you had to do that with anything yet?
There's been a few. I mean, there's been a couple Caliandra species I think we've had, non-orchids, but Caliandras. Yeah, the Triplaris is a tree species that we've, a beautiful flowering plant. You see them all over the place in Miami, but there's males and females, but at some point either males were introduced or maybe there were some ones that had both male and females on the trees, but they start producing seed and
It sounds like a fig issue.
yeah.
You bring the figs in, you want to.
Yeah, but yeah, we certainly actively monitor that, and we have had some orchids reestablished on their own already as well. There's a Cymbidium species that has popped up in some of our Robolini palms. Seems like the Robolini palms end up being a nursery for orchid seed germination, so we do see some of that. I haven't really seen it expand too much further out from like Robolini palms and a couple other palm thatches, but.
So any mulch you produce on property stays on property, you don't.
Yeah, all of our stuff stays on site. We actually have a big giant pile that we actively manage, and so that gets routinely ground up and flipped and turned and made into soil. Nothing leaves the site. And things that, disease or anything like that, we were getting some of the Eulophia graminea popping up everywhere. We would generally just bag those up and throw them away so they would die and not get reintroduced into the mulch, just in case they would regenerate from the bulbs or if there are seed capsules or anything like that on it. We've definitely been excited about the ghost orchids we've put out that have done very well, and it's been a great advertisement for people coming through. You don't have to go an hour deep into the Fakahashi to see a ghost orchid. You can just walk 20 feet off into our boardwalk and see them right there. So hopefully, once these things start hopefully flowering and establishing, and as well as some of the other species that we've put out, I think it'll be a big hit.
I was at the zoo this morning. I was talking to Linda. I can't remember her last name, but she's one of the Collier County School District administrators works in their outreach team, and we're doing a thing there for the school system. And I mentioned I was coming down here for this, and she says, "Oh, well, we've been talking to the botanical garden about planting orchids at some schools, kind of a pet project." So apparently this is still in the development stages, but here is an administrator that knows, in the science department, that knows about what you're doing and knows about orchids, and they're kind of like excited about that. And the reason I bring it up is because, we're a real diverse group of folks here. We represent pretty much what's going on in Collier County. Then we have Atlanta, Fairchild, and Selby, and Smithsonian, and this group's getting bigger, but it's all sort of the same. And the awareness with orchids is growing quite a bit. And my biggest problem with most adults and kids too, but I think kids are going to show us the way in the future, is that they think it's a plant, and if you cut this one down, there'll be another one along, and you'll probably have to spray it anyway because it'll get in the way. And our kids are going to, if you go through this program that Linda was talking about with the school district and start planting some orchids at Manatee, is where she was talking, Manatee Elementary, and they come back in 5 or 10 years, or 20 or 30 years, and see these orchids that are little plants that are probably still going to be relatively little plants in 10 or 20, 30 years. That would be something, because they would learn that even in those small things, there's significance, and I think that's pretty cool.
Yeah, he's touched a number of points there, but certainly, and I'll talk a little bit about that in just a moment, the program, but there's definitely a disconnect. I'm sure we all know a disconnect between nature and plants and children growing up learning about these things. And my wife's a school teacher and biology teacher, and I mean, it's even to the point where kids don't even understand that plants are living things, and it's into that level. So I think, a program like that we're looking into here, you know, a lot of the school districts are just looking for projects, and they're very interested, and Chad might elaborate a little bit more on this, but I mean, they want something like this, and I think we'll be able to provide this, starting with something like an Encyclia tampensis, which in my experience so far from, you know, seedlings that we've grown from seed from our property, they grow extremely fast and easy to grow in the lab. You know, be able to produce these and use these as educational tools. The schools around here, I mean, there's beautiful oak trees and cypress trees in a lot of the schools around here, so having an educational component as well as doing some manual, putting them up into their trees, and, you know, doing programs like this with the school district, I think will be a big hit for us.
You might ask, or Jason, they've got an excellent program going on over in Miami right now with some of the school groups, so
That's definitely
that was fascinating what you guys got going over there.
Yeah, we'd definitely like to learn more. Well, thank you everybody for.
So, the way we do, Jay O'Neill in our lab, he grows in sepia and puts them in little containers with little labels on them, and little folder he gives to everybody at our open house, and it's a huge hit. Kids come back 5 years later, you know, and this thing's about ready to push the lid off, and what do I do with it now? And they actually keep it and really enjoy it. So there's a lot of educational opportunities there. Okay, Larry.
Well, I just wanted to say thank you to Dennis Wiggum, Chad, for your hospitality, for organizing this conference. Time really flies. I was trying to recall when I first started orchid research. Dennis Wiggum and his colleague Hanna Rasmussen invited me to see their operation back in 1992 when I was a graduate student at Clemson. And that's been quite some time ago. So I guess I've been doing this for about 25 years or so. But I wanted to make it clear today that the way I'm gathering this meeting today is pivotal through the lens of history, and it's being recorded. Because what we decide to do today will have a ripple effect for decades in how we approach conservation of our native orchids in Florida and outreaching from there. So I'm going to give a talk today about how we got here through Illinois. I'm actually a Floridian. My father was a professor at University of Florida, a virologist, and he worked with Mike Kane. And so I'd like to come back here. I plan to continue to work here for another 20 years, I hope. And so I'm going to kind of go full circle and weave in a little bit of an international flavor to this, because there's a lot going on in the world that will weave into what we want to do. So just some background information. I am homesick because I live, whoops, shoot.
Forgot it already, huh?
There we go. This is about where I live. It's a long way home. But I've been here about 20 years working at Illinois College. Patty lives further away, look. So about an hour and a half from St. Louis, Missouri Botanical Garden, 4 hours drive from the Chicago Botanic Garden. And Illinois College is rather small, as many of you know. It's, most of you don't know, it's the oldest college in Illinois, 1829. Lincoln actually gave a speech in this building. This was our first, Illinois' first medical school, in fact, years ago. So this building is still here. It's right there. That's Beecher Hall. And then my office and lab is here. We have about 1,000 students, undergraduates primarily. And because I am homesick for Florida, I want to have as many opportunities to come back here as I can. And that pulls me away from the teaching a little bit, but mostly in the summer I come back. So Florida is a very special place because about half of the North American orchid total is found in the state, 106 species or so, give or take a few. And half of those are in this region. And as Nick just said, you're in an epicenter of the distribution of this botanical garden here, in fact. Many of you, we don't have to tell you about this. You're very well aware of this. Mike Owen knows all about this with poaching. But this is what I'd like to highlight, is that the orchids are especially vulnerable because they are intimately tied to the biotic agents that they require to complete their life cycles on their own, to generate spontaneous seedlings on their own, and that includes orchids and fungi. So basically, your lesson to take home today for your class is no pollinators, no seeds, unless it's a rare, unusual, specific case, no life cycle, and vice versa with the fungus. So orchids are masters at exploitation, believe me. But they are also linked so closely to these creatures that they exploit, that if they go down, the orchids go down as well. And Kingsley Dixon and Nigel Schwartz put out a nice article that directly links here. They have said that the more a species is more closely tied to a specific kind of fungus and pollinator, there might be some rarity involved with that, the specialist orchid. So we need to keep this in mind. And orchids are also quite possibly tied to the evolutionary level with their fungi. The student right here, Sarah Unruh, is currently a PhD student at the University of Missouri, and she's using our fungi from Illinois College to develop this wonderful PhD thesis that's currently underway. She will be presenting in Hong Kong in, 3 weeks, I think. So time is running out. Everyone in this room is very fully well aware of this. We have Melaleuca, we have Brazilian pepper, invasive scale insects. We need to get out there and start sampling what's out there, documenting, because this is going to be very different in 50 years. Documenting what's out there and safeguarding the orchid seeds and fungi. Our specialty at Illinois College is to utilize fungi with undergraduate assistants to grow the rarest orchids from seed. We started out with the work I did at Clemson. I brought with me to the Midwest north of the Panthera leucophaea, and has since expanded off into Florida, and now it's in Ecuador and Madagascar. So it seems to be a worldwide need to do this fungus stuff. And so that's kind of our mission statement, is to blend student learning. We're the educational component here and the science component. To blend the education and the science with the undergraduate learning process, because they're going to be the ones to take this on in 20 years when I retire. Like J.J. Sadler there, who many of you know. And since 1996, when I started this at IC, I've had roughly 80 students in the lab. And Lennon Johnson is currently a PhD student at Northwestern through the Chicago Botanic Garden, and he's working on ghost orchid fungi in Florida and Cuba. And so we're going to be seeing a lot more of him very soon, I think. And so Lennon and his other students at IC, they like to do this kind of work for a lot of reasons, but they learn science by doing science. They learn these techniques. They can build up their resumes. They can get into graduate schools a little easier, perhaps. It's always nice to get the young involved with this, because they're going to be the caretakers, obviously. I had 3 students I took to Hawaii, 5, 6 years ago already. And this is a talk I'm going to give in Hong Kong that Jenny asked me to give. And these are some of the seedlings of the rare Hawaiian orchid terrestrial that's found there on the island of Kauai. And we had to put cages up because they release deliberately, I believe, deer, not just pig problems, but deer that hop over fences now, and they had to put these cages in place to protect our plants. But I had 3 IC students with us on this trip, and this was one of the highlights. This was my sabbatical. It was nice to do that. It's nice to work at a small college, because I can open this door where my office is, and the students go to class, and they can come in, they can grow orchids and go back to class and learn all about other things. So this is a snapshot of one of the 2 labs that we have at the college where our students get trained. And once in a while we publish papers. I'd like to point out, let's see, this one is from South Florida, research here. This one is, that one is, that one is. That's the Hawaii one. That's the buffet. The students get their names as co-authors, and that helps them to get the graduate school easier. This is one that we grew, the Epidendrum nocturnum in the Panther Refuge from seed using a fungus that we found. And it did just fine. Scale insect problem down here was pioneered by my student Haley Ray. This is a new one that came out fairly recently, 3 years ago, on Tulasnella that we found in butterfly orchid. Grew it just fine. And the ghost orchid fragrance was investigated by J.J. Sadler and published in that paper. And there's J.J., and he's setting up the sampling apparatus that got to those data. It's nice to get, popular articles out there. It's nice to get the media involved. Honestly, the media is like a sugar high. Once they show up and gone, nobody's going to remember it later. And honestly, those papers are going to linger a lot longer, because science is behind it. So I'd like to point out that the students at the college, look, Dr. King, there's 4 that you might recognize. These are the students that we shipped off to Mike King's lab. Emily, Haley's currently there. He's on her committee, but she's working in the entomology department. There's JJ, here's Scott Stewart, and there's Lamont. Scott is the guy that really is why I'm here. Because when I trained him in my lab, and he worked in doubling the population of Spiranthes breviolabris with a fungus that he used back in 2003. Then he went to graduate school, and prior to going to Gainesville, he worked with Larry Richardson at the refuge, and the two of them kind of got this ball rolling from our standpoint. And then Scott asked me to get involved from here, so that was kind of fun. And there's Emily, she's one of the first students that we sent to the Panther Refuge, and there's 2 others. We send about 2 students down here every July to work for the month. And obviously, I don't have to tell you about this. There's a lot of workers there. They're always, in Illinois, they're always concerned they're going to be eaten alive by a panther. And I have to tell them they're very shy. You don't have to worry about it. But I guess, am I right, Mark? Please tell us that's true. So, it's a lot of fun taking the Illinois homegrown, corn and soybean bred student and bringing them down to this stuff. Hoisting them up, putting them in swamps.
By the way, Larry, I have to interrupt. Jackie and JJ stood in that water or up on the stone all night long. Just like that.
Okay.
All night long. Take
Well,
those out.
makes me feel better about graduating.
Yeah. And they're either going to graduate or die.
Yeah, I remember I spent several hours, 3 hours in the hop ash tree waiting for the pollinator to show up. One day, I got a phone call in my office in Illinois, and a guy had a British accent, but I guess they don't have the accent we do. And it was Dr. Saracen, and he said, I read your paper on the Midwestern orchid, and I'd like you to be part of our research team to go to Madagascar. And your role, he told me, would be to isolate fungi from the island for the first time. And I thought it was a joke. So I said, sure. So I did, and it was quite an honor. But there's a lot of pressure and a lot of weight in the Royal Botanical Garden asked you to do that. So I took part in this project, which we're now into our fifth year here, where we went to Madagascar and to do just that. Actually, Dr. Saracen, I guess, did it as a risk. We went there knowing that we might not come out with anything, but we went, and this was the last trip that we took. And I want to show you how this trip weaves in with what we want to do a little bit. First of all, we're really lucky to be here in South Florida because they have it much worse. They've lost 90% of their forests, and they have a lot more endemics. But 4% of the forest that remains in Madagascar, of the 10% of the forest that are left, it still harbors 4% of the life species in the plant. So there's a lot going on in Madagascar with conservation. There's a lot of lessons that they're learning now that we have the potential to learn from. With the orchids, they have 1,000 species. We have quite a few less here in North America. And many of these, 90% of those are endemic, found nowhere else, like the Darwin's orchid. Seen here. So going to Madagascar was going into an ecological nightmare, and we were trying to gather what's left and preserve it. So this is what we did. We went 3 different trips. Unfortunately, the trip I went on the first time was the year of the locusts. We had a lot of locusts that were flying around and pelting us and looking at us. And then the last time I was there a year ago, we were hit by a cyclone. Now, the reason we went this time of year in January is because we wanted to capture the mycorrhizal fungi that might not be present when we were there during the dry season. Orchids may have a shift in what they utilize during different times of the year. And I'd like to bring that to light here, that those of us that study orchid fungi, we might want to consider this as the time of year as well. So we went during the rainy season, and boy, did we get rain. And so I didn't tell my wife till 2 months later when I got back, but I came really close to death, and I fell into, no, it wasn't this deep, but it was at night. I had no flashlight during the high winds, and I fell over in my backpack. I fell on it, so the energy was absorbed by that, not my spinal cord. But it was a very, very, very tough trip. But that's an interesting thing, is that the road was washed out. We couldn't go any further, so we went to a new place. And we made a discovery. I'm not at liberty to discuss it here yet. But we were turned back by a storm, and now we had a discovery of orchids. And I can tell you that some of the orchids that we got from Madagascar, that work I'm applying here in the refuge in South Florida now, and it's working. And so it's very interesting to see that you can apply the same kind of work here and there and get very similar results. But I wanted to highlight that to do this work internationally, you need permits, and this is just one of 5, I think, maybe 6 we had to get. The U.S. government recognizes orchid fungi as pests. I have to have a special lab just for that at the Illinois College, just for pests. So anytime you bring material overseas through U.S. borders, you have to have permits. You have to have a special place for the fungus to go, according to the U.S. government. Our second trips led to a paper that was just published last year. I can tell you that this paper gave us a lot of different kinds of orchid mycorrhizal fungi from Madagascar, spanning the lithophytes, epiphytes, and terrestrials alike. Lots of different kinds of fungi came out of these. We didn't just took the roots and look at them. We actually pulled the fungus out of the roots and identified them from there to this table. Last month, we used some of those fungi that we got here in this paper, and we tested them on an orchid called Cynorchis purpurea. And it turns out that those fungi that we pulled out of the roots physically removed them and then identified them. Turned out to be very, very effective at seed germination in this first orchid that we tested. We have a number of different orchids that we're going to test. But this is fascinating. This fungus right here, this is the green is the stage 5. Look how many of the seedling total went above the seed viability mark. This was the expected seed viability. That fungus overshot it. So the bottom line is that orchid fungi, if you get the right one, wow. Should I say more? That one fungus. So Illinois College has done some exciting things over the past 20 years, and hopefully the next 20 years will be equally exciting. I'd like to focus, though, on NAOCC, a little bit of Cuba, not a lot. And as Dr. Wiggum said, the Pacific Islands might be. In Ecuador, I can tell you that the Ecuadorians laugh at us in a certain sense because we have about 250 here in North America, excluding Mexico. In Ecuador, we have 40,000. Now, Colombia would argue against that because they think that they have more, but I'll leave that up to them. But they have a lot more orchids in a very small place. And so what I did is I went to the University of Cuenca and met with the president. This was last year, I think, a year and a half ago. And he agreed to fund the National Seed Bank for Ecuador for safeguarding the orchid seeds there. So we're very excited about that outreach. And then recently, as many of you know, we brought in the ghost orchid expert of Cuba, Dr. Mujica, into South Florida. And this is the first trip my sister and I went in 2013. Dr. Mujica is coming back apparently this summer, but he is one of our partners now, and he has documented 100 more ghost orchids in the Panther Refuge than we previously knew. So we're very excited to have him to show us his techniques and how they overlap with Cuba to perhaps get a long-term monitoring system in place. And what we've just found, the data he has just submitted to us, we have now finished off the analysis of the data, and we have this now manuscript that's ready to go. I just have to do a few more things to it. But we've compared ghost orchid populations here in Florida and in Cuba. And let me show you a slide that we were talking about the emerald ash tree borer. Yeah, exactly. I had this on a slide that was presented last weekend. I wonder if you're not right. I'm thinking the same thing. I don't know. But it's something that the genus Fraxus kind of scares me because we know how bad it is in the Midwest. But this is a particularly horrifying picture if you think about it. That's true. And then look how many ghost orchids of the 116 at the refuge are on that one species. So to conclude today, I want to point out Mike Kane's work here has been very important with the ghost orchid research he's done and with the students, and Larry Richardson, who helped get this moving. And with Dr. Wiggins' involvement now, catalyzing the great work that's being done by everyone in this room, I think it is fair to say that if we don't make this work, if the conservation of the Florida's native orchids does not work now, when will it work? And it might be under our watch if it fails. So I'm very pleased and honored that Dr. Wiggins is here to help us let us move forward. This is our goal. And as you just heard, half of that number is here in this part of the world. There's no reason why we all can't get along and get this done. Everybody's got different things they're doing, and thankfully there's a lot of work and diversity out there to do that. So the future, I don't know what's going to happen, but hopefully it will be not staring at the evil witch here. It will be something much more pleasant. Thank you all for your attention.